on YouTube. Right. Hi, Mike. How you doing? We can see you, but we can't hear you.
We can. Yeah. He can talk if he wants, right? He can unmute.
You can, yeah.
Well, as a guest. Yeah.
I've let in people without their mic on because last time it was.
Yeah.
Great. Okay, I am gonna. Okay, now we're streaming. Mike, you can see yourself on YouTube too. Okay. Right, hopefully Andy will join us soon. Hello, everybody. Thank you for joining us for this fourth session. And we have Andy and Grace and Preety, who will be joining Anna Nikolaidou as the moderator. Anna is our program leader for the BSc Architecture program. She is an EU qualified architect, member of the Technical Chamber of Greece, and fellow of Higher Education Academy. She also holds a master's in architecture design from the Bartlett, UCL. She has a broad range of experience in the industry, working for national and international design-led practices. She taught as a visiting professor in architecture at the IE University in Segovia, Spain, and is a senior lecturer in interior design at Falmouth University. Anna's research focuses on Yeah, Anna's research focuses on biomaterials, biosensing, responsive building systems, sustainable design, and smart wearables. She's a member of the Unconventional Computing Lab at the University of West of England. She is currently researching the sensing and decision-making potential of fungal wearables and building systems and their response to environmental and chemical and mechanical stimuli.
Thanks. Great. Thank you, Mirat, for the very, very long introduction. Hello, everyone. Thank you for joining this session. I would like to welcome our guest speakers and our audience to the fourth session of the by Design talk series, Unconventional by Design. This is a session where we will look at how we can design with living material that exhibits not only intelligence, but also decision-making capabilities. I hope that you will find the session interesting, but also provocative, and we can have an interesting conversation. And we have the pleasure and honor to have with us Grace, Chung, Preety, Anand, and Adamanski. So welcome, everyone. We will start the session with a talk by Grace and Preety, followed by a presentation from Andy, and then we can proceed with the discussion and any questions from the audience. So feel free to type in the chat any questions that you might have, and then we can discuss them after the presentations. Great, so I'll introduce Grace and Preety. Grace is an architect. She's currently working for ACME Studios in London. She's a graduate from the AA Design Research Lab, and she has worked for Zaha Hadid Architects and also Heatherwick Studio. Preety is an architect and computational designer. She has worked for a number of globally renowned architectural practices with a focus on large-scale projects set in various contexts. Her key interest is computational development, optimization, and fabrication of complex geometry. Grace and Preety, I will hand it to you now. Thank you very much.
Okay, so yes. Let's start. I hope you can all see the screen. So the chemical machine departs from a critique of current multi-component building systems, which are based on separately fabricated parts incapable of internal communication and self-regulation. These features made us look at an adaptive biological system which is based on environmental information processing for the purpose of self-regulation, which is the slime mode. To embed the behavioral intelligence found in biological systems within architecture, a mathematically equivalent chemical system has been utilized to develop synthetic building components. The technicalities of these theses have been developed by interscientific collaboration with computer scientists, material scientists, and chemical engineers. Our project is also a response to the issue of layered additions of separate building systems and unrelated mechanic systems, where each fulfills limited functions. Rethinking current building systems by proposing an adaptive building tectonic system also causes the critical reevaluation of the role of the architect and all involved technical consultants. The slime mold is a holistic and cooperative biological model, which is based on a small number of chemically reactive members with an underspecified functionality. Furthermore, being an adaptive biological system, it is capable of intercellular communication and interaction with its environment. These types of systems excel in terms of high fault tolerance and systemic robustness. The slime mold has been selected as a subject of study due to its mode of operation. During the plasmodium stage, the organism exhibits behaviors that are described to be intelligent. It appears to be an amorphous yellow mass that develops a network of protoplasmic tubes to efficiently connect itself with several sources of nutrients and hydration. The morphology of the slime mold is highly environmentally driven, and it's computed because of the interaction with the environment. Our research aims to situate the adaptive and information processing capabilities of the slime mold within an architectural context. The behavioral intelligence of the slime mold, including the communicative and regulative mechanisms, are mathematically expressed with the two-variable originator model. The biological modes of information processing can be retraced chemically, as the BZ reaction and the slime mold share the same systemic logic. Therefore, we are implementing chemistry as a synthetic means within matter to embed slime mold intelligence into architecture. The BZ reaction is a model for complex biological systems with coupled feedback loops, which are the base of self-regulation. Despite the difference in constitution and composition comparative to the biological model, the reaction successfully produces visual similarities. On the surface of the solution, patterns visible in nature and variations of the Turing pattern emerge. These patterns displayed because of several operations running at once, reaction and diffusion. We contextualize the mode of environmentally driven self-regulative capacities of the slime mold into the architectural context. We envision a high fault tolerance and robustness in this holistically developed building tectonic system, which is enabled by purposeful chemical communication and morphological regulation. The proposed system is comprised of the environment, the inhabitant, microorganisms, environmental chemicals, and the BZ components. The system is sensitive to environmental conditions of light and carbon dioxide and can adapt to changing environmental conditions. Building services and space generation merge into a seamless whole. To enable communication and inherent self-regulation within architecture, we fabricated components which follow the functionality of the slime mold nuclei. Although there is no direct architectural translation, these components come closest to a building component. Each component is chemically independent and can be seen as the computing engine of the chemical machine. To fabricate synthetic nuclei, the BZ reaction must be materialized to a more tangible solid matter. The synthetic nuclei are made of spherical silicon hydrogel components, which absorb the BZ chemical solution. In terms of materiality, the silicon hydrogel enables reversible morphological change. As the BZ solution can be chemically pre-programmed, specific environmental sensitivities such as to light and carbon dioxide can be achieved at the same time. As per digital simulations, the most suitable component geometry is spherical. Spherical compartments serve the need of sustaining the propagating wave. Digital simulations have proven that the most productive component diameter is 20 millimeters. A decreasing size impacts the capacity to communicate amongst the components. As per digital simulations, a hexagonal packing is the most suitable form of arrangement as it allows the maximum number of neighbors. The arrangement logic also enables the intersystemic nonlinear wave transmission and communication.
So I'll be talking a bit more about some of the physical tests that we had to do in our project at DRL. So in order to physically manifest the digital simulations, we not only had to think about the individual components, but we also had to think about how we were going to encompass them in a form as a system. And we have tested various methods, but mostly with cast membranes, with porous cast membranes. The main challenges that the components needed to be kept physically together to allow physical contact between themselves, which in a way equates communication between those single components. However, the membrane itself also needed to be porous and thin enough for the components to detect changes and challenges of its surrounding environment. We have digitally tested different initial setups and different shapes. So this is no longer in the realm of single components, but as a general formation as a whole. The square is the control, and the circular setup reflects the sectional arrangements, where the circular arrangement maximizes the touch points for each component. And the void in their center is equivalent to the habitable space in section. And this is a test in the spherical section with habitable space in the center, tested with different densities. So although slight, we have found that the reaction is slower when denser, and in a way it is obvious, but the density distribution should also become part of the design consideration. This is a sectional view of our digital prototype, which is mainly composed of two different layers. One which is pressurized, focusing on reacting with the external environment, which will in turn affect the internal layer of components, and the internal layer obviously being more focused to the internal and reactive to the internal environment. And these are physical models that we have attempted to recreate what the internal space and what its look and feel will be like. This was when we first started to play with lighting, as that was the only measure that we could assess within the given measures at the studios at the AA. And we were not only interested in creating a singular space, but also on what would happen if several of these pods were attached to one another. So the digital simulation shows that having more pods could make the system more responsive to outer environment. This is not necessarily in the rate of reaction, but rather the amount in which it reacts with its surroundings. And obviously having a greater number of reactive components exposed is reflected in the simulation result. And this is the Arduino setup for the lighting test that we actually implemented. We have attempted to create a feedback loop where LEDs would feed back into the light sensors, and also the environmental light would feed into the light sensors. So in order to simulate the script in the physical world, we initially tested it with the actual BZ reaction that you can see in the bottom right-hand corner. This is when you truly realize the great divide between the physical and the digital. The BZ chemical reaction requires constant perturbation of some sort, and once the chemicals reach equilibrium, the reaction dies. And it only lasts for a few minutes at the longest. And however, please be aware that at the AA we couldn't access a full lab setup at the time to carry out these tests. So since the setup beforehand with the actual chemical didn't quite work, as we showed you the previous slide, we've set up a similar setup, but instead of where the hydrogels were encased in the plastic membrane, however, due to the fickle nature of the BZ solution, we have attached it to the processing script that simulated the BZ reaction. This simulation is as per diagram shown previously, and due to the presence of light sensors, you can actually see switching lights on and off. We were actually switching the light on and off in the actual room to see how physical prototype reacts to environmental changes. This is a cyclic diagram that shows how we see this loop sits within our diagram, our design. This is a system that is fundamentally different to the current architectural practices of division among architects, engineers, consultants. And this is a project where such division is no longer applicable, and where the role of, say, architect or the designer needs to be redefined with those who give technical guidance in achieving which we now call architecture. Yeah. And this is actually the full image of what I've shown as previously as a section. And this is the rendered view of our prototype deployed on Mars. And this is the view of our final simulation based on the initial 2D simulations. And this is describing the three-dimensional space. We were envisioning a non-static space which could expand and contract in accordance to its use or its non-use, and reacting sufficiently to hard environments and providing adequate protection for its inhabitants. Yep, so this is the interior view. Yep, thank you. And that's all from us.
Thank you. Very interesting and inspiring project. Thank you very much. We will now proceed with Andy's talk. Andy Adamatzky is a computer scientist, well-known, international acclaimed researcher. He's a director of the Unconventional Computing Lab at UWI and a professor in unconventional computing. He specializes in designing computer models and experimental laboratory prototypes of computing devices made from non-standard substrates. He has an astonishing record of funding collaborative projects and publications, and it's an honor to collaborate with him on several projects. Andy,
okay, so I will just say thank you. I will share my screen now. Yeah, I will try.
Yeah, thank you.
So now I will try to somehow remove it. I just checked. Do you see like how I'm changing slides? Because sometimes it doesn't work.
Yes. Yeah.
Yes, okay. Yeah. So I will give a brief talk about fungal neuroscience and the sensing and computing with mushrooms. So basically, what you see on this picture is spiking behavior. And looking at the picture, we couldn't say which is spiking behavior of neurons, real human neurons, or actually mushrooms. And if you go to the next slide, we can see that somehow it's dropped anyway. Yes. Go on the next slide, we will see like basic characteristics of the action potential, which is common for neurons of nervous system of mammals. It exists of the depolarization phase, action potential per se, repolarization, and refractory period. And previously it's thought that such spiking behavior is only kind of known for nervous system and for living creatures which has actually which has brains, or at least ganglia, such as snails, for example. However, about 3 years ago, I decided to check what we will see if we just stick electrodes into the fruiting bodies of mushrooms. And I have done several experiments, and I found that basically if we insert one electrode in the stalk and another electrode in the cup of the mushroom, then we record spiking behavior extremely similar to that recorded from neurons. So we have, as you can see in this plot, depolarization phase, repolarization, and refractory period. So basically, mushrooms, they spike as neurons do. And that's examples of fungal spiking. We have very distinctive spikes, which have the same parameter apart of width as neuron spikes. So there are several kind of types and sets of spikes. There are slow trains of spikes, where spike can be like about 1 hour in width. There are trains of the fast spikes, where spike width can be 5 or 10 minutes. And mushrooms basically communicate by the trains of spikes. So what we try to understand, we try to kind of infer if the spiking behavior of mushrooms somehow could be mapped from a set of stimuli. And therefore, we kind of established a new field of fungal sensors. So here I will just provide a few examples. First one is fungal skin. If we grow mycelium in a very particular manner, liquid culture, so it will be like floating on the surface of the supplement materials, then fungal skin is produced, which is very similar in structure to the skin of animals, as you can see on this picture. If we have certain electrodes in this living fungal skin, we will record spikes. Spikes can be singular spikes or trains of spikes. If we apply load to this skin, as you can see on this plot, W is the star, load applied and W all load removed, then this fungal skin reacts with a large spike and then like wave train of smaller spikes, decreasing in the amplitudes. If we apply light, the fungal skin responds with increasing overall potential, and then it keeps this potential until light is on. Therefore, we can see that fungal skin discriminates between types of stimuli. Between a pressure stimuli, it just on-off, and light stimuli, it stays on until the stimuli actually is removed. Okay, and in parallel, we developed another project with unknown reactive fungal variables, and the idea was to check if actually we can colonize mycelium on our kind of clothes, and if this clothes will react to some physical and chemical stimulation. So what we have done, we colonized, we took a hemp tissue, fabric, and colonized it with oyster mushrooms. You can see mycelium is white on this photograph, and then we insert electrodes, 8 pairs of differential electrodes, into this hemp fabric colonized by mycelium, and recorded electrical potential. To check on the how it reacts with the stretching of the fabric, we attach this hemp part to the real t-shirt of the mannequin, and then we are applying weights to the actually bottom of this t-shirt. So that's photo how it was actually attached and how electrodes were arranged. And the whole mannequin was covered with some plastic to kind of reduce evaporation and keep hemp basically humid. So what we found, if we apply good stimuli, like for example malt extract, then we have a response with a high oscillation of spikes, as you can see on the right of the plot, high oscillation of spikes. If we apply ethanol, which is actually very bad because it damages mycelium, we can see a response in the very high amplitude spikes, and then all spiking activity is actually extinct. So mycelium doesn't really like it. If we start to stretch a t-shirt, which actually part is attached by weight like 50 gram, 450 gram, we will see responses with singular spikes. Exact kind of exact sign of the spike doesn't really matter because it depends on how waves of excitation pass between electrodes. But reaction is actually very strong. So this our fungal variables can detect also stretching as well. Another way to check pressure was to use fungal blocks, as you can see on this photo, fungal blocks, sizable ones, and we can insert electrodes into the sides of the blocks, and then apply some weights. Well, here on the photographs, you can see we applied 16 kilograms. And the fungal blocks response to the application, you can see on it's 16 kilo was actually applied, and off 116 kilo is removed. And they respond with single large amplitude spikes, and they respond with strains of low amplitude spikes. And what we found that actually we can discriminate in the plots of the spike amplitude versus width. We can discriminate after the application of pressure and after the removing of pressure, because you can see on this plots index of the fit, linear fit of the plot, they are different. They are different. So basically, these fungal blocks discriminate between weights. And what also we found interesting, habituation. So if you repeat stimulation many times, then response of the fungal blocks will be less and less. This habituation is very common in living systems, especially in living systems with central nervous system. Right. And what other experiments we can done? We checked if it's possible to make computation with fungi. That was done in simulation. We simulated single fungal colony. We insert matrix of electrodes. We stimulated the colony with by injecting current. You see propagation of the current on the colony on this snapshots. And then we kind of agreed that depending on the which input string was entered in the substrate, one or another type of logical gate is actually constructed, as you can see on this photo. And different color means different input strings. So 0, 1, 1, 0, and 1, 1. And then we calculate frequencies of the logical gates implemented. That's, you can see how the spikes are recorded. And that's distribution of logical gates. So AND gate or OR gate are most common, which is typical for other living system. And XOR gate, this XOR gate here at the bottom, is very, very rare, which is also common for other living systems like slime mold or plants. So basically we can compute with mushrooms as well. And the last point, we were curious at the moment to ask, are fungi conscious? And at the conference on consciousness like 2 years ago in Switzerland, we asked Stuart Homerov, who is father of studies of consciousness, actually what does consciousness think? And he responded, if a thing can be anesthetized, the thing has consciousness. So we decided to check if mushrooms can be anesthetized. So we used chloroform, and it was published in this paper in bioarchive. And we actually took fungal substrate, and we applied chloroform in the container with the substrate, not directly, but in the container. So we have a vapor of chloroform. On the application of chloroform, spiking activity either was completely diminished, as you can see on this plot, or it was diminished at first, and frequency of spikes was reduced, and amplitude of spikes was also reduced. But then, when chloroform was removed, after some period of 6 to 12 hours, when all vapor actually gone out of the container, the spikes start to return. And you can see on the right part of the plot, and especially in the zoom part of the insert, how actually mushroom actually wake up, spiking returns, and amplitude becoming larger and larger. That's basically that's all. And if you have any questions, please feel free to ask. Thank you very much.
That's great. Amazing, Andy. Thank you. I will kick off the discussion by asking a few questions, and then we can answer any questions from the audience. You're all invited to type your questions in the chat, and we can have a chat, even if there are any comments or anything you would like to mention. For years, we have been looking into living material and living organisms, and we're trying to emulate and mimic the processes, the behavior, performance, efficiency of those living organisms and living material in order to provide solutions to design problems in different fields, in architecture, product design, and in many different fields. And I have a feeling that we are starting to move in an era, a post or meta biomimicry, biomimetic era, where we're not looking only into nature, but we're working with nature, we're working with living material, and we're taking advantage of the extreme potential and capabilities of the living material in order to design. And I would like to invite you, Andy, and Grace and Preety to comment on what are the benefits, but also what are the limitations of using biomaterial, for example, slime mold or fungi, because you've discussed intelligence and sensing, being able to sense environmental or chemical, mechanical triggers, and their ability to adapt or maybe take decisions. What are the benefits? And since it's a material, it's a living material, what are the limitations that we have when we work and when we design with, for example, fungi or slime mold?
Just before we start the discussion, we just wanted to also acknowledge Andy was also a great supporter of our project while we were in DRL. In terms of living materials, for architecture at something at that scale, it is always a great challenge. The unfortunate thing is that if you look at the more prehistoric or very primitive forms of architecture, it's in a way much more living than the architecture that we live in today. And this whole real shift sort of began as people started to use concrete and things which were in a way almost unnatural to us in our built environment. I think for architecture and anything at that scale, it's scaling up that is the problem. And because architecture or any sort of building or a home has also the function, will always have the function of protecting humans from the outside environment or from wild animals. So there is a certain amount of robustness which is required or it's mandatory. So I find it's a bit of a technology issue in terms of scaling up. And even if it's soft, if there is something that is robust enough for people to inhabit, perhaps that's where the future lies, and something that is more responsive to not only to us, but also to the environment surrounding us.
Yeah, that's great. Preety, Andy, I don't know if you have it.
I would like to just add something to this based on our project. So we were not actually looking into mimicking the physicality as a visual or morphological aspect, but we always wanted to mimic or embed the intelligence, as we've mentioned during our presentation. And that is not something, at least we believe, not something you could achieve by just using the mold as a building material, because that is also not following practical reasons, as Grace already explained. It's not, I mean, there's not the amount of experience or the amount of technical expertise to use it directly as a building material, but we were also never really interested in like using it as it is as a material, because it doesn't really allow for the thermal properties or the structural properties a material needs to have. And we were actually, since we were always looking into the microscopical level intelligence, which is found on another level, it's not found on the level you actually see the material. It was always clear for us that, you know, we want to mimic the intelligence and the behavior, which does not necessarily require us to use the mold, but to use its behavioral intelligence. And that's why we decided to use a synthetic mode of embedding that intelligence. So yeah, it was never really an option for us to basically use the material.
Use the material as it is.
And also not mimic the physicality, but actually mimic the intelligence, learn from its capacities and capabilities.
Andy, and maybe I will extend my question and maybe we can discuss can we actually use, if we use a material as it is, the biomaterial, can we actually take advantage of its capabilities of sensing? For example, can we use it as biosensors for our cladding, for our buildings, or for our wearables, to sense different environmental variables such as pollution, let's say, or light, or changes in temperature? So how can we design intelligent sensing systems in smaller scale and larger scale? And I agree with Grace, scaling up is always a problem because it requires quite an advanced degree of technical manipulation of the biomaterial. So yeah, Andy, if you have anything to comment on that. But also, what are the limitations in terms of, we are working with living materials, so in order to keep it alive, we need to, provide water. So could it be, for example, a good alternative for exterior cladding systems where you have rain, or how we can actually take the work from a lab setting into actual real life applications?
Yes, many questions. I will try to answer one by one. First, yeah. Advantage because living structures can self-repair, therefore they actually can grow by themselves. Second, because living structures can feel the same as we feel, therefore it will be easy to integrate between inhabitants and actually buildings. Third, we can embed intelligence inside building materials because, for example, if we make a building with a living mycelium, then these fungi mushrooms will actually sense and make decisions by themselves. That's actually advantages. Disadvantage is that we should keep these living materials alive. And to keeping alive is always challenge, especially systems like, well, chemical systems are not safe really like Biosensors reaction because concentration of malonic acid is quite high. So we can just ignore it at the moment, talk about mushrooms and slime mold. And all these two kind of species, they require high humidity. Therefore, to make building incorporating slime mold and fungi, we must somehow encapsulate them in semi-permeable membranes so they can breathe and transfer oxygen and water, yeah, for example.
Yeah.
But the biggest challenge is actually language, because indeed living materials can sense, indeed living materials can make decision. However, we must know how to talk to them to understand what they're saying to us. And from my point of view, it's now biggest challenge at the moment. Yes.
Yeah, I agree with you. And one other element that I would like to bring to the discussion is, can actually create structures or create, for example, living skins, both working at a smaller scale, such as wearables, for example, or larger scale as buildings, that can actually communicate with each other. Because if we found that fungi produce electrical spikes and there is electrical activity when they are exposed to different variables, can we actually establish communication patterns, not only between the skin with the environment or the skin with the user, but between the users or between our buildings? And I think that might sound a little bit, forward thinking, but is it something that can be done in the future, and we can, work with communication between the skins as well?
Yes, it can be done because communication can be done not just with physical contact, but via electromagnetic fields or even optically. It's well known that all cells, they emit photons. And there are even recent research telling that neurons in the brain also communicate with photons. And therefore, in principle, in future, we can communicate with living materials just by light, just recording what photons they emit and then sending back photons. That's my vision at the moment.
It's great. Grace, Preety, I don't know if you would like to comment anything on this one before we proceed.
There was also, we actually, in our thesis, which was a very thick book, there was also a small bit that we touched on about this, because if we are to change, I think actually building out of living material is the final frontier. It is the final frontier. But at the same time, in order for that to be achieved, it also means that us as humans need to overhaul how we use how we live in the environment that surrounds us, because how we live now is unsustainable, potentially very different to living in an architecture that is built out of living things.
Yeah, that's great. And I agree with you. Oh, Preety, yeah.
No, I just wanted to add something that during our thesis, the aspect of controlling a building system was a big topic, like, because right now we have this deliberate control where we turn on the light and turn off the light. But if you have a living system or an intelligent material, which is actually your building, which is actually part of your built environment, then the mode of control, how you control that system, is actually going to be quite different. And that's the aspect of language, which Andy touched on. What is the language, or what is the control, or how do you control that system? Because it's not something like, you're not turning on the light, or you're not turning the switch, or whatever. It's actually going to be a different way of communicating between the user and the built environment. And that's also a challenge. How do you, and that will also in effect have an impact on how we live and how we use our built environment. That's kind of going to change our habits, our mode of inhabitation, and it will have a big impact on our day-to-day life.
Yeah, very interesting. And this leads to my next question, which is, and I can see that we have a few questions in the chat. I'm going to go to these after this question. Can we actually make predictions if we can map the spiking activity, the electrical activity or output from the living material when it's exposed to different environmental, chemical, for example, or mechanical triggers? Do we have is there potential to make predictions about, for example, the amount of pollution that our building might be exposed to, or the temperature, or the light, or even chemicals, and how we can train or use computing or machine learning, for example, if we can, to do that, to make any predictions?
That was question to me.
Yeah,
okay.
I think so, yeah. But everyone is very welcome. I know, Andy, you are the specialist on that. So.
Well, so far with mushrooms, we are not sure at the moment. We're just studying language of mushrooms. Based on my experience with slime mold basically react to the good stimuli with low frequency, high amplitude oscillations, and bad stimuli, high frequency, low amplitude oscillations. That's the whole language of slime mold we understood after 10 years of studies. But it's still good. And hopefully with mushrooms, we also understand that there is some kind of very rough mapping between set of stimuli and set of reactions. But we shouldn't be particularly optimistic about that one, because take, for example, cats and dogs. We live with cats and dogs for hundreds of thousands of years, and we still don't understand their language. So, and we communicate with them, humanity, on a daily basis for so long time. So we shouldn't expect to understand language of slime mold or fungi just in 2, 3, or 5 years. It will take a substantial period of time. But we should stay optimistic. Eventually we will be able to speak with mushrooms and plants.
Great, thank you. Okay, I will go to the first question from our audience. You talk about these materials being exposed to the environment elements as if they would sit as an outer skin on a building, but do you see it as potentially easier in scaling them up to consider them for roles inside an existing building where they are offered some protection? Grace, do you want to go first, or Preety?
Do I understand the question correctly, that we should integrate that system or that living or synthetically living material into existing buildings? Would that be correct, if I understand that correctly? I guess that's what is meant, but I think that would perhaps solve a few issues like structural issues or longevity issues of that living material. Maybe it would be a way to start that process, and then to further, with that experience gained by using it within built structures, you could then gradually move to the built environment fully built by living materials or synthetically living materials. I think that would be an option to integrate it first into existing structures and see how that would change our environment and how that would change the way we live. And then gradually we could move on to fully applying this material, I would say.
Great. Andy, Grace, I don't know if you have anything to comment on that.
Interestingly enough, when we actually did our physical test with, like literally those balls that you saw were like hydrogels in this sort of skin. One of the reasons why we decided to deploy to Mars was the lack of gravity. So basically, hydrogels tended to be, or at least anything that had some form of a liquid embedded in the material was obviously heavy. But we wanted it to somehow be a bit more, let them be able to move and react a bit more. So literally we stopped looking within Earth. We started to look beyond. To answer the question, the fundamental design was that the whole entire system will be created by a singular or very minimal types of material. Like that's the actual starting point of our whole project. Maybe we might be able to apply, but I guess gravity would eventually get the best of us, I think. Yeah.
Okay. Andy?
And honestly, I didn't think about that one, so
yeah.
No one else.
It's okay. We can go to the next question, which is actually addressed to you, Andy. How can living biosensors be incorporated into computers?
Well, how they can be interfaced. If the question was how they can be interfaced with the normal computers, then they can be interfaced optically or electronically. Yes, because in any case, biosensor just changing its physical state, either by spiking, change of electrical activity, or by changing shape like sine mold. So it will be optical actually inputs. So electrical and optical interface. If question was how to integrate with computing, then computing actually is embedded into these materials. So decisions are made immediately at the stage of accepting stimuli. It's like protocognition. It's very similar to olfactory bulbs, which we have in our nose, because some stimuli, some molecules recognized already at the level of the nose before actually they reach brain. That's how I think it will be, computing will be integrated.
Great, thank you. I don't think we have more questions in the chat. Feel free to add any comments or anything you would like to ask our guest speakers. I have another question which relates more to the fabrication or biofabrication of structures or skins, maybe to the body scale or building scale, so smaller or larger scale. And do you see, or what type of fabrication methods or processes can we actually implement, and how we can move from using the fungi in the lab into real life applications, which I think is, it's a big step that we need to overcome. And maybe I would bring into the discussion 3D printing with living material, if that's an option. I know we're trying to work on that, Andy. So yeah, if you would like to comment anything on the fabrication processes with fungi, for example, or slime mold or any living material, and what are the potential of that?
I think when you start fabricating with biological materials, there's always the aspect of unforeseen changes and the lack of control you have. And also you kind of want to have some sort of standardization, like you want to know how that material will behave or what will happen under certain circumstances. That is the reason why we chose those synthetic nuclei, because we were able to actually control parameters such as scale, such as chemical concentration, and it is kind of a synthetic system where we are deliberately programming its behavior. That was one of the reasons why we chose not to use the actual biological material, because it's very hard to foresee the future behavior. But yeah, as you mentioned, there are some tests being done with mycelium 3D printing to embed, to develop a material where the actual fungi is used as a component and to actually use its behavior or its properties within an actual material. So that is actually something very, very interesting, but it's not what we were thinking back then when we were doing our thesis.
Yeah, great. And I think this leads to the next question from the audience, from Fidel. He's asking, do these organisms have freedom? Can they decide to act in a different way and rebel against the mathematics and logic, against what mathematics and logic expect? And if so, is there any ethical dimension in manipulating them? And it's not a joke, that's a comment. Andy, do you want to answer?
Well, all living creatures react by laws of physics and chemistry. Therefore, kind of, they do have freedom, but in the sense that everyone has freedom. We just react by our biological laws. And about ethics, well, difficult question basically, because if you think about human body, actually it's just a consortium of millions of cells, and each cell very egoistic and they have their own agenda, but they still stay together and they keep us alive because it's in common interest basically of the all cells for us to stay alive. The same with our interaction with living materials, for example. Indeed, we can do some unpleasant things to them, but it will be for the common interest of all of us staying alive. That's my vision.
Great. Okay, so a question from Pat. If a building is built from living materials and controlled by artificial intelligence, will our homes start to replace our pets as living companions? Grace, do you want to answer that maybe? Or Preety, Andy?
I think the question is also situated in the notion of how we live now. So, you know, we have a static home and a pet, which is obviously more moving and, you know, emotionally attached to us. But we really don't know what that would be. What would living in a living house or a living building would be. But it would be different. I mean, yes, I mean, it could be an issue of control and how we control the environment, but it could even potentially be a completely no control, where we don't have to control anything because it is almost environmentally reacted and controlled on our behalf. Yeah, I mean, I don't know whether I'm really answering the question, but yeah.
Yeah, great. Great. I would like to ask, until we're waiting for any more questions from the audience, please feel free to type your comments or questions. But I would like to ask, what are the applications? What is the impact? And also the fields that could benefit from the use of biomaterials. Yeah, so maybe, Preety, do you want to answer that? Since this MSc, the Masters that Mirat is setting up is also addressed not only to architectural students, but also, you know, students from different fields. And I think it's an interesting question of where do we go from here and what are the applications, what fields can benefit from the use of biomaterials and their intelligence and sensing capabilities, decision-making capabilities, but also their sustainable possibilities because they can self-grow and they can self-repair, they're abundant, you know, in nature.
Yeah, I think for us, like at the moment, the biggest or the most important application would be the built environment because it makes up a very big chunk of the environment, let's say. Mm-hmm. And if we would, and also there's a huge amount of building waste being created, and there's not the recycling building material, there's no closed cycle. So it would be like, being an architect, I would obviously say that the built environment would be one of the biggest benefiters of this kind of a shift. And it would also mean that different engineers and technicians would start working together, and it would really enhance the multidisciplinary working methodology. And it would also be very, very helpful for developing sustainable systems. So, I mean, this is very generic what I'm saying, but I would say yes. For example, the built environment would benefit. I think the computing industry would benefit, because while we were doing our research, we also came across a lot of computers which are now not like with a fixed body plan, but they're actually built based on biological or chemical systems, which is kind of the next revolution, coming from the digital and electrical revolution going to the chemical revolution. So it's kind of an industry which could also benefit a lot from these systems. And also, of course, as you are also researching on the clothing industry, perhaps could also benefit from biological materials, as the recycling or the reuse or the re-implementation could be a lot easier than with synthetic materials.
Yeah, I agree with you. And I think so far we have been almost considering the user of the building and the building as separate entities. But I think maybe with the use of biomaterial, we can bring these two entities together. And for example, we have been working on a project with Andy on hormones. Can actually fungi sense hormones? Hormones are released by our body when we are, for example, in stress state. So, you know, according to our emotions, our body, you know, fluids change. So can actually the living material react to the user, or the emotional state of the user, or even detecting disease? Or can the building do that actually? You know, can you touch your wall and they can sense, you know, they can sense the user, which I think is something, you know, that might seem very, you know, forward thinking, but
it is radical. And that's why our project was also like called radical in many ways, because it would actually, what would it mean if a wall, for instance, starts sensing or it starts making decisions on our behalf? We don't know. And that's like the radicality of this approach. And even if it's embedded within computers or materials or whatever, we don't know what the implications will be. And our habits are not based on this kind of a material. So it's going to be a big shift in how we use our environment and the materials surrounding us.
Yeah, great. Andy, would you like to comment? Or...
Yes, I would just to echo the same was said, that in fact we already have living materials, because first of all, our digestive system has about 2 kilograms of microbes, so basically lining. And second, on our skin, we have also millions and millions of bacteria, basically covered by bacterial biofilms. Therefore, we are already living with this living building materials. We're already covered. And when we make real buildings also living, we will just extend these living materials slightly further, in the same manner as spider using web as extension of itself. We will just extend ourselves. Yes, that's my vision.
Great. I can't see any questions in the chat. Oh, there is one. Can the mycelia used as an electrical wire, or is it not necessary as long as it is living? Andy, do you want to go for this one?
It can be used as a wire, but it will not live forever. Therefore, it can be used as a wire only for like short period of time, few days or weeks. Yes.
Great. I would like to ask a question which is more of a technical question that refers to the process of designing, since we have students who are attending or will be watching this session. And my question is, how do we start, or where do we start? So for example, do we bring into the conversation the design process and designing, or do we identify problems in the design process and then we investigate the appropriate material to provide solutions to that problem? Or do we start with the biomaterial and then the design process is guided by its properties or capabilities that exhibits, or both, for example? And can we actually be creative when we are using living materials in the design process? As architects and also all designers, there is always this element of creativity. So how restrictive it could be the use of biomaterials, and also for the students that will be starting working first time with biomaterials, where do they start? How do they start? Nature provides like a huge databases with different living material, living organisms that they can look that they have homeostatic responses, they are adaptive. Where can they start? And I know it's more of it's not a specific question that has like right or wrong, but maybe we can have a chat on that for the students who are joining the course and they are wondering of how do we select our material and where do we start in the process of designing. And maybe Grace, I don't know if you want to go first.
I mean, I think there has been a big push, especially within the last 10 years or so, for actually using more biological, or at least biologically friendly, or at least environmentally friendly and sustainable materials in architecture as well. And the push has been, the push has always been sort of there, but I think it's really became a big agenda within the last decade. With the architecture, the tricky part is the fact that it is related to a lot of money. It is related to a huge industry of commerce and real estate. So for any, I would say, any form of new material or a new method of construction to be rolled out, it really needs to be somewhat of, whether it be at least academically verified or research-wide verified material for it to even be considered for it to be applied on a larger, more commercial scale. But having said that, when we approached our project, what we were suggesting wasn't just a different type of architecture by itself, by our project in itself, but a different way of inhabiting space and potentially a very different structure of how we actually build. So it's not, more and more, it's really just the developers who are just pumping in money to build in. I mean, they're not going to be in any way, shape or form be interested in any form of new materials, unfortunately. And in order for that to really change, there needs to be a greater shift. And I think it's coming, and it's inevitably coming. Yeah.
Okay.
I mean, I would like to add something to the aspect of design. So, I mean, most architects, they have a very traditional way of designing the building. Like, it's very much top down. It's very intuitive. It's based on sketching a space or a building. And then you just see what materials could you use as per the concept or as per the most suitable application. But with this, like also speaking from our thesis, we actually developed the material first. And we did a very, very long time of research in order to understand the biological system and then the chemical system. And it was a more scientific approach. So if architects were to use biological materials, they would need to become more scientific, I think. They cannot design the way they were designing until now and then just apply the material. But the material itself requires its own way of designing in order to work within the environment and within the setting. So that's also something we wanted to really express within our thesis, that form of material has its own aesthetics, it has its own design rules, and only then it can function within the setting we are proposing. So it was a very bottom-up approach for the design, and it was more like we are providing the setting, we are providing a setup, the material composition, but the actual, it's very, very little amount of intuitive top-down design. So it needs to have its own setting in order to work. So it will definitely also have a shift in how architects work. It will be a more scientific and a more collaborative and a more interrelated and collaborative approach to designing. And it's not that you have the design and then choose the materials, but the material will also guide you through how you need to design it. And that was the difficulty during our thesis, that we had a material,
yeah,
we had done the research, but then the design part was really hard because we basically started from the end, and then we kind of started the designing process. So it will have to have a shift.
The issue, I think, partially is that it actually starts from first year architecture, because you learn functionality of a building as a separate subject to aesthetics. And if you look at our natural systems, the aesthetics is actually achieved by its function, and it's not a division. And a lot of the architects at the moment still practice that aesthetic side of things rather than, what is functional. But when you say functional to them, it's a very different idea of a function, not something that is integrated and combined.
Yeah, I agree with you. Great. I don't see any other questions. So I think, Mirat has a question.
Yeah, I'm trying to type it like everybody else, but my computer is freezing. So I'm just gonna ask. I have a two-part question that goes. Some of it goes to Grace and Preety, and the other one goes to Andy. But it's mostly about simulation or determining the parameters or some sort of indices before you start investigating. And so on the architectural side, with Grace and Preety, the simulations that you're showing, especially the one on the interior where you're assuming on the inside the space is moving as it's being used, so it's a dynamic space. I was wondering, were there any other architectural or spatial logics in the simulation that define because the camera seemed to have not been out of the camera seemed to be always in the space. And so I was wondering, there's probably more parameters in that. And then on the flip side, which would be for Andy, is when you're using these materials to test them out to see if they can be used as gateways, or you can create these logics with these live materials, do you run simulations before, or do you, and what might that be? Or do you actually just, essentially probe these things and see what happens?
Okay. Grace and Preety, maybe you want to go first?
Yeah, on the simulation, it's an interesting point that you pointed. Yes, you're correct. The camera angle was actually stationary. The space is closing in on you because the space is no longer needed. So what we mean by that is that when the space no longer detected a certain period of inhabitation, so nothing to react to, then basically the space is closing in on you. So that's why we were actually suggesting a completely different, almost monetary system of dealing with architecture within our project. Because imagine, with real estate, you buy a 3-bedroom house and whatever, but in our case, it's like you just buy a house. It may have 3 bedrooms if you need it, but if someone's no longer in that other room, then that basically room would close in on itself and it's no longer there. So it's a nightmare for estate agents, I would say. But you're right. The reason why it fluctuates is the general idea, the big idea that we had behind was that if it's used, it'll either maintain, or if it detected more people, perhaps it'll get bigger because of the more CO2 and more weight or more people touching and changing the pH of the actual components. And then there's an enlargement. And then after following a certain duration of static, where there's no perturbation or no manipulation, then eventually the space would close up itself.
Yeah.
So
I mean, adding.
Sorry. So there is an. I'm gonna let you respond. Sorry, but there is that is one of the things that is really important because a lot of people are listening are architects, and it would be really important to explain that there is architectural indices into that. And Preety, I guess you're gonna say that. Sorry.
No, sorry. What did you? I forgot what I wanted to say, but
Sorry.
Just adding to that, it's also one of the things we were suggesting is that you can't have a fixed architecture if you actually want to have this really strong connection between the inhabitant, the environment, and the system. It would be wrong to define an architecture. But the question was always, so what is the design? And it is kind of, it doesn't match anymore to design something very determined and very specific and then propose this system. So that was also what Grace was describing as the change in its arrangement or in its spatial arrangement or in its volume, which is dependent on the habitation. So how do you design something like that? How do you determine, and why would you determine something like that if it's actually going to adapt itself? So that was also like one of those questions raised during our thesis.
Great. Andy, I don't know if you have to reply with.
Yes, I remember the question. Yeah, the question was, do I do simulation before? No, usually I don't, because we only need simulation if you couldn't do things physically. If you can just take mushrooms and start recording, it will be much quicker to just start recording, because there is no such model which reflects all behavior of real system 100%. Every model is just approximative. Therefore, we only need modeling when we couldn't do things physically.
Great, amazing. So I think we can wrap up the session. If there are no questions in the chat, which I can't see any. Yeah, so I think we can wrap up the session, can complete the session. I would like to thank our audience and the people who are watching on YouTube as well. Our guest speakers, Grace, Preety, and Andy. It was amazing to have you in the session. And Mirat, back to you.
Okay. Can you guys hear me? Because Zoom crashed.
Yeah, we can hear you.
Okay. Apologies for those on YouTube that it crashed while Anna was. Sorry. Technical difficulties. Anyway, what I'm trying to say is thank you, everybody. Thank you, Anna. Thank you, Andy. And thank you, Preety and Grace. And I guess we're going to end this session just quickly because everything on my computer is freezing.
Yeah, it's freezing. Okay, we can hear you now.
Okay, can you guys hear me?
Yeah, we can hear you now. Now you.
Okay. Right. So, thank you, everybody. I'm going to make this quick. Final Friday is Research by Design. We will be... our guests are Stefania Soldini from Liverpool. Can you guys hear me? And Liam from Squid Soup. They are. I will be moderating that session. Hopefully, if my computer doesn't die within the next 5 minutes. And thank you, everyone.
That will be. Okay. It's breaking up a little bit.
Yes. But I'm hoping, luckily it didn't break up with all with the good stuff. So there's that.
Yeah, we didn't hear very well, Mirat. It was breaking up.
Okay.
I don't know if you can repeat what you said.
Okay.
We can hear you more clearly now.
Right, hopefully. Okay, next Friday, Research by Design. Our guests are Stefania Soldini from Liverpool University from the Space Dynamics Lab, I think, and Liam Liam Brittles from Squid Soup in Bristol. And I think I'm done. Thank you. Thank you very much.
Great. Thank you. Thanks, everyone. Thank you.
Right. Thanks. That was.
Thank you. Bye. Bye.
Thank you.
Thank
Andy,
you.
very
Thanks,
much.
Andy.